good evening to all of you. How are you? Please write in the box. Am I audible to you? Let's start with the new topic, that is liver function test, which is included in Veterinary Analytic Biochemistry. This is Unit 3, Analytical Biochemistry. Am I audible to all of you? Yes, please write in the box. Yes. How are you people? Okay. Hope you all are well and fine. Today's topic is liver function test. In the previous class, we have studied about the clinical enzymology, means the study of various enzymes which are diagnostic important or importance is of diagnosis purpose. Let's start with liver function test. This is also an important clinical aspect of this analytical biochemistry, that is liver function test. You all know liver is the largest gland of our body and animal's body. So this is a central body for the various functions like metabolic functions, excretory functions, synthetic functions, very important detoxification and storage purpose. These are the major functions of liver. This is the largest gland of our body, and also it is the central this is the central part of the body which correlates or regulates metabolic function, excretory function, synthetic functions, detoxification and storage. This diagram shows the various functions of liver which are very, very important, like cholesterol synthesis. This is a very important feature. You all know this, the conversion of glucose into glycogen and again glycogen into glucose. What is this? We call it, please write in the box, conversion of glucose into glycogen. Please write in the box, glucose convert into glycogen. We can call it, yes, glycogenesis. And further, glycogen converts into glucose. It is also called, it is called as, yes, please write in the box. When glycogen converts into glucose, it is called as glycogenolysis. First is genesis and other is genolysis. You all know. We have already studied the carbohydrate metabolism. Next step is, next function, this is deactivation of poisons and toxins. We will study in detoxification function or genobiotics or metabolism of genobiotics. We will discuss it further. This is amino acid synthesis. This is hormones, enzymes production. You all know enzymes are biocatalysts. They are proteins in nature. The production of hormones and enzymes and detoxification. Bile, we all know, gallbladder. This helps in the absorption of vitamins. This is the synthetic process, means analytic process, and degradative means catabolic processes. So you all aware by the functions? Next is, what are the main functions of liver? Although we know, but just summarize. Metabolic functions. First is of carbohydrate metabolism, glycolysis, you all know, glycogenesis, you all know, glycogenolysis, gluconeogenesis, then conversion of galactose and fructose, galactose metabolism, synthesis of galactose, then blood glucose regulation by various hormones. So glycolysis, glycogenesis, glycogenolysis, gluconeogenesis. What is gluconeogenesis? Please write in the box. What is gluconeogenesis? Please write in the box, what is gluconeogenesis? Am I audible to all of you? Yes, what is gluconeogenesis? Yes, gluconeogenesis is the synthesis of glucose from non-carbohydrate moiety or non-carbohydrate compound. Next, the protein metabolism, protein catabolism, you all know urea cycle, amino acid metabolism, and hormone synthesis. Fat metabolism includes fatty acid breakdown, oxidation, you all know. Oxidation of fatty acid is called as, please write in the box. Fatty acid is called as, yes, Shipra, yes, please write down. What is the oxidation of fatty acid? Name of the oxidation of fatty acid. Yes, this is called as beta oxidation of fatty acid. You all know. Next is, you all know citric acid cycle. TCA is Krebs cycle or tricarboxylic acid cycle. And another is ATP synthesis, or we can say oxidative phosphorylation. You all know about this citric acid cycle and ATP synthesis, which is included in biological oxidation. Yes. There is some. Yes, am I audible to all of you? Again, I am asking. Yes. Next, function is related to synthetic part, that is anabolic in nature. First is protein metabolism, synthesis of plasma proteins like albumin, globulin, coagulation factors like fibrinogen, prothrombin. Please write down the some coagulation factors. Please write down in the box some coagulative or coagulation factors or factors which are useful in blood coagulation. Please write down. Number 1, factor 1. Which is factor 1? Please write in the box. You have studied earlier. Which one is the first factor for the coagulation, blood coagulation? Yes. First is fibrinogen. You all know? Yes. Likewise, prothrombin, then proacetylene, and many others. Stewart factor, you all know. So, liver is the main organ for the synthesis, which is essential for the synthesis of various coagulative or coagulation factors like fibrinogen, prothrombins, and for various enzymes also. Next, fat metabolism, synthesis of cholesterol. TG, you all know what is TG? Neutral fat. Triacylglycerol. Triacyl. What is the another name of triacylglycerol? Please write in the box. Triacylglycerol or triacylglycerol. What is another name for this triacylglycerol? Triglyceride or TG or neutral fat. Okay. Next, various fatty acid synthesis and lipoproteins, digestion of lipids with the help of various bile salts. We will discuss in detail about bile acids and bile pigments, bile salts. Next is very important hematological function. Liver is also produces various clotting factors. I just told you, like factor 5 and factor 7. Factor 5 is pro-acylserine and factor 7 is pro-convertine. Fibrinogen, you all know, involved in blood coagulation, is also synthesized by liver, and it synthesizes various plasma proteins, and it is also responsible for the destruction of RBCs or erythrocytes, which is very important function of liver. Excretory function is very important, as metabolic or synthetic functions like bile, pigment, bile salts, and cholesterol are excreted in the bile into intestine. You all know about the process. Next is protective function, or we can say detoxification. Another name for this detoxification is metabolism of genobiotics. This is a very important word, genobiotics. Ammonia is detoxified to urea. You all know ammonia is converted to urea. Then Kupffer cells, liver, Kupffer cells, where it is present. Kupffer cells, you all know, in the liver. Kupffer cells of liver perform phagocytosis to eliminate various foreign compounds or foreign particles, or we can say various. We will discuss further about the metabolism of detoxification of drugs, alcohol, and various hormones. Liver is responsible for the metabolism of genobiotics, that is called as detoxification, that is removal or removable of excretion of drugs, alcohol, hormones, and bilirubin. We will discuss in detail about the metabolism of genobiotics in next class. Storage function, that is storage of glycogen, iron, vitamin A, D, B12, or trace element like iron. ADEK, vitamin A, D, E, K are, you all know, these are fat soluble. What is LFT? Definition of LFT. In the simplest words, we can say test to assess liver functions. Test to assess liver functions. In detail, we can say the liver function test, LFT, common name, are the biochemical investigations to assess the capacity of liver to carry out any of the function it performs, like metabolic, excretory, detoxification, storage, and synthetic functions. LFT will help to detect the abnormalities and extent of liver damage. So what is LFT? Please write in the box. Definition of LFT. Please write in the box. What is LFT? Am I audible to all of you? Yes, am I audible? Pankaj Kumar. Yes, please. Write in the box. Pankaj, write the definition of LFT. What is LFT? Am I audible to all of you? Do you hear me? Okay. What is LFT? Please define. Please define. Test to assess the liver functions. Liver is very important central organ for the various functions like metabolic, anabolic, detoxification, storage, and excretory. So, what is the importance of liver function test? It is important. Or we can say the clinical aspect or clinical applications of liver function test. First is it helps in the diagnosis of liver diseases, categorizes the liver disease into various categories, or we can say differentiate type of liver diseases like jaundice, hepatitis, obstructions, cirrhosis, hepatoma. There is a little difference between all of them, so it is very important. Following the progress of liver disease, or it is important degree of parenchymal cell damage. What is the degree of cell damage? It is important for the clinical aspect of liver functions. It judges the prognosis of patients, and it also evaluates the response to the treatment, like therapy with statins to check hepatotoxicity and therapy for various coagulation disorders. So it is very important clinically. It is very important because it also denotes the diagnostic feature of various diseases. In simple way, we can say it helps in the diagnosis of various liver diseases or hepatic diseases. Classification of liver function test. It is very, again, very versatile. According to function of liver, you can enlist it. Test based on excretory function, test based on synthetic function, test based on metabolic function, test based on in storage function, and test based on detoxification or metabolism of xenobiotics function. So group 1, based on biochemical findings. You all know what is biochemical findings. What is biochemical findings? We can judge the various biochemical factors like in blood, serum, plasma, in urine, etc. So first group, test based on liver excretory function. Serum bilirubin, you all know. Serum bilirubin, bilirubin and biliverdin. What is this? Serum bilirubin and serum biliverdin. Please write in the box. Bile salt or bile acids or bile pigments. Please write in the box. What is serum bilirubin? Yes, bile pigments. Okay, very good. These are bile pigments. So serum bilirubin can be judged by or can be diagnosed by Van der Berg reaction. You all know about this. We will discuss it in detail when we will study jaundice in later slides. Serum bilirubin by Ictidis index, urine bilirubin and bile salts, urine urobilinogen and fecal stercobilinogen. Urobilin and fecal stercobilin. And last one is for the assess of excretory function of liver. This is bromosulfethylene B.V.Sc test. Bromosulfethylene B.V.Sc test. Next group is liver enzyme panel test or biomarkers for liver injury or cholestasis. We have already studied these parameters in our previous class, that is of clinical enzymology. Please write down what is SGPT. What is another name of SGPT? Please write in the box. This is totally repetition. What is SGPT and what is SGOT? Yes. Alanine transaminase ALT and aspartate transaminase SGOT. Next is alkaline phosphatase. You all know gamma glutamyl transferase, that is GGT. Why GGT is important? This is the biomarker for which disease? Please write in the box. Alcoholism. Yes, right. Serum isocitrate dehydrogenase. Next is cholinesterase and 5-nucleotidase, that is called as 5-NT. So these are the various liver enzyme panel which helps in the diagnosis of various liver injuries or diseases, or we can say cholestasis. Next group is group 3, test based on liver synthetic functions. Plasma protein concentration, you all know in serum, total protein, albumin, globulin, serum Ag ratio. What is this serum Ag ratio, albumin, globulin ratio? We will discuss it in further classes. Fibrinogen concentration, then flocculation tests in amino acid in blood and urine. Test for blood coagulation. This is known as prothrombin time determination. Prothrombin time determination. This is the test which is done for blood coagulation. Group 4, tests based on liver specific functions. These are very important, like alpha-1 antitrypsin, AAT. These are plasma proteins, globulins. This is a biomarker of hepatitis, alpha-1 antitrypsin. Next is alpha fetoprotein, that is also called as AFP. This is a biomarker for tumors of liver and germ cells. Ceruloplasmin, you all know, and ferritin. Ceruloplasmin, alpha-1 antitrypsin, alpha fetoprotein. These are increased in this liver hepatitis, or we can say tumor of liver and cirrhosis. Test based on metabolic capacities. First is galactose tolerance and antipyrin clearance or antipyrin breath, and test based on detoxification. You all know hippuric acid synthesis. We will take it in further slides. This is the another classification which is based on clinical aspect. Markers for liver dysfunctions like serum bilirubin. You all know von der Berg reaction, serum bilirubin by Icterus index, urine bilirubin and bile salts, urine urobilinogen, fecal stercobilinogen, urine urobilin and fecal stercobilin, serum total protein, albumin globulin and serum AG ratio, that is albumin globulin ratio, which is very important for various diseases, prothrombin time determination, blood ammonia when indicated. Group second, markers for hepatocellular injuries. Enzymes, you all know, serum glutamate pyruvate transaminase, SGPT, is also called as ALT, serum glutamate oxalate transaminase, SGOT, this is called as AST. Group third marker for cholestasis, you all know, ALP is important, or ALP, alkaline phosphatase, is the diagnostic marker for cholestasis. And gamma glutamyl transferase is also a marker for cholestasis and as well as alcoholism. Am I audible to all of you? Yes, am I audible? Please write in the box. Rahul Ujwal, am I audible? Okay. Neha, am I audible? Yes. Neha, please write in the box enzymes, name the enzymes which are important for the diagnosis of cholestasis. Neha Jange, please write in the box enzymes which are important for the diagnosis of cholestasis. Neha, am I audible to you? Suresh? Yes. Suresh, please write in the box enzymes which are important for the diagnosis of cholestasis. Suresh Kumar Mehla? Priyanka? Am I audible to you, Priyanka? Priyanka Meena? Kaleem Ahmed? Okay. Am I audible to you? Please write in the box. Nisha? Next is formation and excretion of bile pigment. You all know by the fragmentation of RBC or destruction of RBC, we will get free bilirubin, bilirubin glucuronides conjugated, and then various compounds like urobilinogen, stercobilin in intestine, and in kidney you also know this urobilin, and then it excretes in the urine. This is the whole process of formation and excretion of various bile pigments. Name the bile pigments. Please write in the box. Am I audible to all of you? Yes, bilirubin and biliverdin. Liver function test based upon bile pigment metabolism. First is, you all know the constituent of bile. Bile salts, bile pigments, and cholesterol. Am I audible? Okay. Bile salts, bile pigments, and cholesterol. LFT in serum based upon bile pigment metabolism are serum bilirubin and biliverdin diazosulfonic test, or we can say van der Berg reaction or van der Berg test. Next is for estimation of serum cholesterol by various methods. Icterus index, we will discuss in later slides. And for LFT in urine and stools, urine bilirubin, gambling test and Foucher test. These are the two tests which are very important for a diagnosis of bilirubin in urine. Gambling test and Foucher's test. We will study in practicals about these tests. Urobilinogen and fecal stercobilin by LH test, and urobilin and fecal stercobilin by Sklenizer's test. Bile salts. Bile salts. Name 2 bile salts. Please write in the box. What are these bile salts? Please write in the box. First is sodium glycopolate. So always remember these are bile salts, and bilirubin and biliverdins are bile pigments. So don't confuse. These are the bile salts, that is sodium glycopolate and sodium taurucocolate. What are the functions of bile salts? Emulsification and addition of lipid, you all know, absorption of fat soluble vitamins, cholesterol solubility, and what are the liver function tests for bile salts? Hayes sulfur powder test. You all know about Hayes test? Please write down. Hayes test. What is Hayes test? Hayes test. We have performed in the practicals. Hayes sulfur powder test. Hayes test. What is Hayes test? And another tests are Pitton-Coffer's test or liver peptones test. The commonly used test is Hayes sulfur powder test. Have you heard the name of Hayes sulfur test? Please write in the box. Van der Berg reaction. For the serum bilirubin, this is a reaction between bilirubin and Harich diazo reagent giving a reddish purple compound. What is conjugated bilirubin and what is unconjugated bilirubin? Conjugated bilirubin reacts directly with the reagent. This is called as direct bilirubin. It is important. Direct bilirubin is also called as conjugated bilirubin. What is unconjugated bilirubin? This does not react with the reagent directly except after addition of methyl alcohol. Conjugated bilirubin gives purple color within 30 seconds, and unconjugated bilirubin does not react with the reagent directly except after addition of methyl alcohol. Thus, it may be called as indirect bilirubin. So, conjugated bilirubin is also called as direct bilirubin, and unconjugated bilirubin is also called as indirect. So, please write in the box, what is indirect bilirubin? What is another name of indirect bilirubin? Please write in the box. Am I audible to all of you? Kalpna, what is indirect bilirubin? What is the another name of this indirect bilirubin? Sujata. Okay. Mohit, what is direct bilirubin? Mohit. Rahul Kataria. Vinita. Yes, am I audible to you, Vinita? Okay. So, conjugated bilirubin is direct bilirubin and unconjugated bilirubin is indirect bilirubin. This table is very important. This is a biochemical, this indicates the biochemical changes for the differential diagnosis of 3 types of jaundice. First is hemolytic jaundice, second is obstructive jaundice, third is hepatic jaundice. Hemolytic jaundice is also called as prehepatic, obstructive is post-hepatic, and hepatic is also called as intrahepatic or hepatic. So hemolytic is prehepatic in nature. Obstructive jaundice is post-hepatic in nature. So we can see here serum bilirubin. Here in hemolytic jaundice or prehepatic jaundice, unconjugated bilirubin is increased, and in obstructive, conjugated increased, and in hepatic jaundice, both. So it is called as biphasic, both conjugated and unconjugated. So Van den Berg reaction shows indirect positive because the increase in unconjugated bilirubin. Direct positive, obstructive jaundice because there is increase in conjugated bilirubin, and biphasic means both unconjugated and conjugated bilirubins increased. Serum enzymes, what is the status of these serum enzymes? ALT, AST, ALP, normal. In obstructive jaundice, these are normal in hemolytic jaundice or prehepatic. Obstructive jaundice, ALP increase, ALT and AST marginal. ALP, that is called as ALP. What is ALP? Please write in the box, what is ALP? Please write in the box what is ALP. ALP, am I audible to all of you? In obstructive jaundice, which enzyme is increased? Alkaline phosphatase. Okay. What is ACP? Please write in the box what is ACP. And this is the biomarker. ACP is the biomarker of which disease? That is acid phosphatase, and this is the biomarker for prostate carcinoma. You all know. We have studied earlier in our previous class in clinical enzymology. So here in hemolytic jaundice, ALT, AST and ALP normal. In obstructive, ALP is increased and ALT and AST marginally is increased. In hepatic jaundice, this is the important point here, ALT and AST increased and ALP marginal increased. In urine, we can see in urine, what is the status of bilirubin in urine? It is not excreted in hemolytic jaundice, but it is excreted in obstructive and hepatic both. And what is the status of urobilinogen in urine? This is excreted in hemolytic, but may or may not obstructive jaundice, and you can say normally obstructive jaundice and hepatic may be decreased or it may be normal. So this is the important aspect of this differential diagnosis of three types of jaundice. First is hemolytic, second is obstructive, this is post-hepatic in nature, and third one is hepatic, it is intrahepatic. So you can see here unconjugated and conjugated both bilirubins are increased in hepatic jaundice, and in obstructive conjugated only, and in hemolytic unconjugated. So this shows indirect positive von den Berg reaction, and this shows direct positive von den Berg reaction, and this is biphasic in nature. ALT, AST, ALP normal, ALP increased, and here ALT and AST increased marvelously. Then bilirubin not excreted in obstructive and hepatic, and urobilinogen excreted in hemolytic jaundice. Now it is clear to all of you the differential diagnosis of three types of jaundice. Am I audible to you? Please write in the box. This is clear to you? Yes, please write in the box. This is clear. Now this is clear to you? Okay. Let's go to the next slide. Yes, this is the important feature. This is the crux of these differential diagnosis of jaundice, von der Berg reaction and jaundice. If it is indirectly positive, so we can diagnose it as hemolytic jaundice. What is hemolytic jaundice? Another name of this hemolytic jaundice? Direct, yes. Next is direct positive. So it diagnosed, or it will diagnose as obstructive jaundice, and if it is biphasic in nature, it is hepatic jaundice, or it will diagnose as hepatic jaundice. So now it is clear to all of you the differential diagnosis of these three jaundices and the reaction. Okay. Yes, you all know about these biomarkers of impaired liver functions. These are the normal functions like heme, catabolism. This is increase in bilirubin in the case of impaired functions. Enzymes, ALT, AST, and GTT, all are increased. Protein synthesis, always remember this albumin is always decreased. Mainly in chronic conditions, and this prothrombin time is increased. Urea and ammonia increased. Cholesterol, triglyceride increased, and half-lives of drugs, because it involves, liver involves in detoxification of drugs. So here half-lives of drugs are also increased in the case of impaired liver functions, and this is called as this is increased in bile acids also. So important point is albumin is decreased. Here albumin is decreased and all other parameters are increased. Please remember this albumin is always decreased in the case of impaired liver function. It is particular in chronic cases. Now it is clear? Yes, abnormal protein electrophoretic patterns in liver diseases. So in the case of acute hepatitis, prealbumin, or which is also called as transthyretin, is reduced. Then cirrhosis, albumin reduced. I just told you cirrhosis, in cirrhosis, liver cirrhosis, albumin reduced, gamma globulins increased, albumin decreased or reduced, but globulins may be increased. Hepatocellular diseases, alpha-1 globulins reduced parallel to albumin, and this biliary obstruction, alpha-2 globulins or beta globulins increased. The rise in gamma globulins will have wide bases, and this is suggestive of polyclonal gammopathy. We will study in detail in further classes abnormal protein patterns in various diseases. So don't worry, we will discuss in detail in our next classes. There you have to remember only this: albumin is reduced in liver cirrhosis or hepatitis, in chronic cases particularly. Yes, what is the pattern of transaminases in liver diseases? You all know parenchymal liver cell damage, plasma ALT and AST increased. In viral toxic hepatitis, plasma ALT and AST increased. Please remember, ALT is always present in cytoplasm and AST is present in both cytoplasm and mitochondria. So increase in ALT is greater than AST, where the cytoplasm sustained major damage. And in case of cirrhosis, liver cirrhosis, you can see here increase in plasma AST is greater than ALT, where the cytoplasm and mitochondrial membrane are damaged. In active hepatitis, ALT and AST normal or slight increase. Cholestasis, you all know, slightly AST ALT increase, but in case of cholestasis, ALT, ALP, and GGT increase. ALP is alkaline phosphatase. ALP is the biomarker of cholestasis. In alcoholic hepatitis, ALT and AST ratio reversed, and in cholestasis, ALP and GGT also increased in alcoholism. So the normal AST ALT ratio, the AST ALT ratio is AST upon ALT is increased greater than 2 times in myocardial infarction, alcoholic hepatitis, and cirrhosis. You have to remember these: alcoholic hepatitis and cirrhosis. In the case of alcoholic hepatitis and cirrhosis, AST is more increased than ALT. So AST ALT ratio is increased 2 times greater or more than 2 times greater. AST ALT ratio is decreased. It may be called as ALT higher. So AST upon ALT means ALT higher and AST low. It is present in acute hepatocellular damage and cholestasis. This is the important point here. This is AST upon ALT ratio is decreased in acute hepatocellular damage and cholestasis. And this AST and upon ALT, AST upon ALT ratio is increased. AST increase or ALT low. In case of, you all know, the biomarker of myocardial infarction is AST. AST is the biomarker of myocardial infarction. Please write in the box the earliest biomarker enzyme of myocardial infarction. Please write in the box with its isoenzyme. Only CPK? No. It is CK2 or creatine kinase or creatine phosphokinase 2. So now it is clear, this AST upon ALT ratio, please add in the box. Is it clear to all of you? AST ALT ratio, AST upon ALT, AST increase, ALT low. Here AST and ALT ratio decreased means AST low and ALT higher. Okay. Alkaline phosphatase, ALP is an indicator of biliary obstruction. You all know obstructive or post-hepatic jaundice. ALP is also elevated in cirrhosis of liver and various hepatic tumors. You all know gamma glutamyl transpeptide, this is called as GGT, is highly elevated in biliary obstruction and alcoholism. 5 nucleotidase, that is 5-NT, is elevated in hepatobiliary diseases. And this LDH, what is LDH? Please write in the box, what is LDH? What is LDH? Am I audible to all of you? What is LDH? Lactate dehydrogenase. These isoenzymes, LDH5 and LDH4 are useful as LFT. It is useful in liver diseases. So, one by one, we will discuss the various tests. First is prothrombin time determination. This is important LFT or liver function test. Prothrombin time required for clotting to take place in citrated plasma in which optimum quantity of thromboplastin and calcium have been added. So here you can see prothrombin, thrombin, fibrinogen, fibrin clot. Important point is this: prothrombin time increases in hepatic and obstructive jaundice. You have to remember this line only. Prothrombin time increases in hepatic and obstructive jaundice. Next test is galactose tolerance test. What is the procedure of galactose tolerance test? You can see here 40 gram of galactose given orally or 350 milligram of galactose per body kg weight. We give IV solution. Then we will take the sample fasting and every half hourly over a period of 2 hours. Then compare with the normal galactose level. In normal individual, 3 gram of galactose appear in urine within 4 hours. It is just like a glucose tolerance test. 4 hours after oral supplementation and blood levels decline progressively at each fasting level by 2 hours. So in liver disease, it is important here. In liver diseases like cirrhosis and hepatitis, galactose utilization decreased. And urine excretion of galactose is more than 3 grams. So here galactose utilization is decreased. It is the important point. It is decreased in cirrhosis and hepatitis. Likewise, excretory functions. The test is for excretory function, bromosulfathine test as LFT based on excretory function. Liver performs function of excretion of drugs and dyes. Here, bromosulfathine dye is used. Intravenous injection of bromosulfathine dye, 5 milligram per kg body weight as 5% solution. And this is safe to use in humans. And we can say here, it gives purple color in alkaline medium. So dye retained in liver diseases. It is important point. It is not excreted, so it denotes the abnormal excretory function of liver. Now it is clear to all of you. It denotes the abnormal excretory function. As we can say here, dye retained in liver diseases 50% almost. So is it clear to all of you? This is bromosulfathine test. This is a dye based on excretory functions. Here, dye retained in liver diseases up to 50%. It indicates the abnormal excretory function. Now it is clear to all of you. Is it clear? Am I audible to all of you? Do you hear me? Okay. Hippuric test or hippuric acid test. It is important. It is associated with detoxification or metabolism of xenobiotics. It is associated with metabolism of xenobiotics. Detoxification process. What is hippuric acid? You all know. Benzoic acid, which is toxic in nature, plus glycine. This is conjugation reaction. Hippuric acid forms. So here you can see a reduction in hippuric acid excretion, particularly less than 3 grams, indicate hepatic injuries. There are several processes like precipitation of hippuric acid with ammonium sulfate and dissolved precipitation in water, and then titration with NaOH. Ultimately, the result is reduction in hippuric acid excretion indicates hepatic damage. It is important. A reduction in hippuric acid excretion indicates hepatic damage or abnormality of liver. In next slide, you can see the test, which is again related with bilirubin icterus index. You all know about this Icterus index. This is a simple test to measure yellow color of serum, which is due to the presence of bilirubin. It is often useful in rapid assays. So this is the formula: potassium dichromate solution. It is the formula Icterus index, volume of diluted serum and volume of serum used. It is important in neonatal jaundice or physiological jaundice. It is absolute. Now this test is not performed as much. Absolute test. Next is aminopyrin breath test. Aminopyrin breath test. It involves measurement of 14 CO2 formed from methyl labeled aminopyrin in liver by demethylation. In liver diseases such as cirrhosis and hepatitis, the amount of this 14 CO2 is decreased. It is important point here. CO2, this is labeled with 14 carbon, is decreased in liver diseases. What is the limitations of liver function test? I have tried to cover all the important liver function tests. This is the limitations of liver function test. First is extrahepatic or extrahepatitic factors, means except liver, there are many factors like nutrition and all. They affects the liver functions. Enormous reverse capacity of hepatocytes. You all know they have the capacity of regeneration of parenchymal cells, and various tests lacks sensitivities. So these are the limitations of liver function test. Is it clear? The limitations of liver function test? Yes. There are many extrahepatic factors, regeneration of parenchymal cells, and various tests lack sensitivity. They are not accurate. So what is the conclusion of today's class, which is about LFTs, liver function test? Therefore, diagnosis of liver disease should be based on history, histology, and biochemical findings. A combination of laboratory investigation. It is an important point here. It is a combination. We suggested a combination of laboratory investigations are employed in LFTs. These include serum bilirubin, conjugated and unconjugated, ALT, AST, alkaline phosphatase, GGT, and proteins like albumin and globulins. So this is the conclusion of today's class. That is, we prefer the combination of various laboratory investigations for the LFTs or for the liver function itself. Am I audible to all of you? Okay. So, Nitesh Chaudhary. Nitesh, do you hear me? Nitesh, please add in the box. Yes. Yes. Please add in the box, Nitesh. Do you hear me? Sachin Kumar. Sachin. Sachin. Do you hear me? Sachin. So, now I repeat the whole class. Rajendra Singh. Please add in the box, what are the major functions of liver? What are the major functions of liver? Please add in the box. Rajendra Singh, Rahul, Sachin, Jitendra Kumar, please add in the box. What are the major functions? Yes, detoxification. Of course, detoxification. What is genobiotics? Please add in the box. Genobiotics. Rahul, please write down. Please add in the box. What is genobiotics? Metabolism of genobiotics. Now, I repeat the whole lecture in 2 or 3 minutes. So, liver is a very important organ or central organ for various functions like metabolic function, excretory functions, synthetic functions, detoxification or metabolism of xenobiotics, and storage. Here, metabolism of xenobiotics important because removable or remove of excretion of drugs, alcohol, hormones, and bilirubins. It is important. It is essential. What is the definition of LFT? Test to assess the liver function. So LFTs helps to detect the abnormalities and the extent of liver damage. This is the importance of these liver function tests for the diagnosis of, for the prognosis of, and for evolution of the response of the to the treatment of therapy or treatment or therapy. This is the classification of liver function test. Test based on excretory system, test based on synthetic, all are based on the function of liver. These tests are important. One, the Berg reaction, Icterus index, and this B.V.Sc excretory test, it is called as bromosulfethine test. These are the various enzymes: SGPT, SGOT, ALT, GGT, cholinesterase, 5-NT, and these are the tests based on plasma protein concentration and blood coagulation. This is prothrombin time determination test is important. Time is increased. These are liver specific tests. These are plasma proteins like alpha 1 antitrypsin, AAT. These are globulins, alpha phytoproteins, ceruloplasmins, and ferritin. These are liver specific proteins. Tests based on metabolic functions, galactose tolerance, antipyrin clearance, and this hippuric acid synthesis for the detoxification or metabolism of xenobiotics. What are the bile salts? Sodium glycocholate and sodium taurocholates. Bile pigments are bilirubin and biliverdin. These Derberg reaction, this is a very important reaction for diagnosis of jaundice. Conjugated bilirubin is direct bilirubin, unconjugated bilirubin is indirect bilirubin. This table is very important for differential diagnosis of three types of jaundice. This is hemolytic, prehepatic, obstructive, posthepatic, hepatic, intrahepatic. It is important here. Indirect positive is for hemolytic jaundice. Direct positive is the diagnostic feature of obstructive jaundice, and biphasic is for hepatic jaundice. Here you can see in hepatic ALT, AST increased, and here in hemolytic ASP, ALT, AST, ALP, these are normal. So indirect positive test indicates hemolytic jaundice, direct positive test indicates obstructive jaundice, and biphasic is for hepatic jaundice. These are the biomarkers. You have to remember this albumin, it is decreased in impaired liver function. Here it is important again, AST-ALT ratio. AST-ALT ratio is increased in MI, that is myocardial infarction, alcoholic hepatitis, and cirrhosis. AST-ALT ratio decreased in hepatocellular damage and cholestasis. ALP, you all know, increased in obstructive post-hepatic jaundice, and GGT, alcoholism, obstruction, and 5-nucleotidase is elevated in hepatobiliary disease. Likewise, LD5, LDH5, LDH4 isoenzymes are useful as LFTs. These are some tests: prothrombin test, galactose tolerance test, bromosulfethine test for excretion, for excretory function of liver. This is hippuric acid test for detoxification. This is Icterus index. It is a diagnostic test for serum bilirubin. And these are the limitations of liver function tests like extrahepatic factors like nutrition and many others, enormous reverse capacity of hepatocytes, regeneration of parenchymal cells, and some tests like sensitivities. And this is the conclusion or suggestion that a combination of laboratory investigation are employed in LFTs like serum bilirubin, both conjugated and unconjugated, ALT, AST, alkaline phosphatase, GGT, and various plasma proteins like albumin and globulins. So this is all for today. Thank you very much. So thank you again